Hey, ladies and gents, welcome back. I'm sorry for my abrupt um, ending to the previous video. I'll need to speak to the director of all this. So anyway, now we're going to get into organelles and look at what all the different organelles do. Organelle just means little organs. So they're these little tiny structures, most of them membrane bound, that have specific combinations of enzymes in them, and they do different jobs. So again, increase efficacy or efficiency here. So let's start on the outside and look at plasma membranes. Plasma membranes, again, it's a phospholipid bilayer. So you have two phospholipids or two layers of phospholipids with their polar tails or polar heads on the outside and non-polar tails on the inside. So what it does is it helps control what can enter and leave the cell. Again, it's semi-permeable. It allows some things to pass through readily, but not others. So it allows cells to control what goes in and out. And again, it gives them capacity to sort of have their own metabolism. Let's see. Next, we have the nucleus and ribosomes. The nucleus is a large organelle in the center of the cell, and of course, it is important because it contains the genetic information. It contains DNA wrapped around these histone proteins in a molecule that's referred to as chromatin. So, chromatin is DNA wrapped around these proteins. It's sort of remarkable. You've got about 6 linear feet of DNA in your 46 chromosomes. If you fully unwound them and laid them end to end, it would be as long as a table. It'd be 6 feet long. But they wrap up around these histone proteins, so they pack neatly into the nucleus. Also, within the nucleus, during certain stages of the cell cycle, you can see a dark dot in the center. That dot is the nucleolus, and the nucleolus is functionally important because it's where ribosomes are made. And of course, ribosomes are super important because ribosomes are like the little chamber where polypeptides get made. You link together amino acids and make polypeptides that will fold up and become proteins. So the nucleus is all about protein production. Now, what we're going to look at next is the endomembrane system. The endomembrane system combines several different organelles into sort of a process. I think a lot of times when people initially learn about organelles in the cells, they learn about individual organelles and the jobs they do. In reality, those organelles sort of work in concert with one another to carry out a number of different functions. So we're going to be looking at the endomembrane system. It works with the rough and smooth ER, the Golgi, lysosomes, these little transport vesicles, which are like little pouches that can move around and transport contents or materials within a cell. So you can sort of see how there are all these membranous sacs, and again, we'll have different things going on inside of them. So let's begin with the smooth ER. The smooth ER gets that moniker because it doesn't have ribosomes. So it looks smooth under a microscope. No, the smooth ER, again, it has these pouches or spaces. It's called lumen. Lumen is just another word for cavity. So within the lumen or the lumens or the cavities of the smooth ER, there are enzymes that do things like make phospholipids and nonpolar hormones. So the smooth ER is like a membrane factory. So membranes are made or produced within the smooth endoplasmic reticulum. Another cool feature is some of the cisternae or pouches in the smooth ER have enzymes for detoxifying different harmful substances. So you can have the smooth ER be sort of an indication of um, uh, abuse of like alcohol or drugs. If a person, has a lot of consumption of alcohol, one thing that can happen is the detoxification that can happen in different organelles. So the prevalence of certain organelles can be an indication of prevalence of use of different substances. Another interesting thing is that you can add hydroxyl groups. You can add these OHs to toxic substances, and that increases their solubility. So if they dissolve, then what happens is the waste products or the toxins will pass into your bloodstream and then get filtered out by your kidneys. So you can detoxify certain substances, break them down, or you can also make them more water soluble so you can flush them out of the body. And we'll also look at instances where those pouches in the smoothie can actually store calcium ions that actually plays a role in cellular communication. We'll talk about that more in the next unit. Now, the rough ER is rough because it's got ribosomes. It's studded with ribosomes. And we know ribosomes assemble polypeptides, right? Ribosomes are involved in protein production, so you can guess that the rough ER is about making proteins. So these are secretory proteins. In a lot of cases, they're proteins that get made and then released from the cell, and then will go somewhere else in the body. What you can sort of imagine is the rough ER is membranous, right? And it has the ribosomes. So the ribosomes can make little polypeptides that fold up, and then you get vesicles bubble off. You get little pouches bubble off to transport these proteins to other parts of the cell. And that's where the Golgi comes in. Sometimes people call the Golgi like the shipping department and stuff like that. What will happen with the Golgi is, again, it has these pouches called cisternae. And within those pouches are enzymes that fold up and activate proteins. Some of the proteins are going to be exported from the cell, like what you see in this image. So again, this is really nice. You see the rough ER. Here are little purple proteins being made inside of that. A vesicle pinches off, travels to the Golgi. Inside the Golgi, the shape will be changed in those proteins and they'll be activated. And then they can be secreted from the cell and go do something like, be insulin to help lower blood sugar. Again, the Golgi has enzymes for modification and packaging. I like the little modified design for the Spotify logo here with the Golgi apparatus. So let's watch a clip where we see this in action. I hope the audio worked for you guys. Sounds like a soap opera. It's very dramatic. I feel like you have to watch my reaction with this. It's very awkward.
It is important that translated proteins are delivered to their specific cellular location. To accomplish this, the protein is transferred through a series of membrane structures. A principal membrane component is the Golgi apparatus. The Golgi apparatus is the sorting organelle of the cell. Proteins from the rough endoplasmic reticulum are sent to the Golgi. As the proteins move through the Golgi apparatus, they are modified and packaged into vesicles.
You can call it
because the Golgi apparatus receives proteins from one location and targets them for delivery to a second location, it is sometimes considered the post office of the cell. The Golgi apparatus consists of general components.
It's got its hours cut.
The cis cisterna nearest to the endoplasmic reticulum, or ER, the medial and trans cisternae, and the trans-Golgi network. Other key players in this process are the proteins being transported and the enzymes that modify them. Translated proteins are encapsulated in vesicles in the ER.
So the proteins get made and packaged up.
And these fused vesicles form the cis cisterna. As the protein moves through the stack, it is modified by resident Golgi enzymes at specific locations in the apparatus. These modifications are important because they provide the signal that determines the final destination of the protein.
Okay, so you
how does the
activate
protein move through the Golgi? Movement occurs in waves. First, the cis cisterna becomes part of the medial Golgi cisternae. Behind it, a new cis cisterna is formed by the fusion of vesicles from the ER. Meanwhile, one of the medial cisternae migrates and becomes the new trans cisterna. Collectively, this process is known as the cis maturation model. Proteins are sorted within the trans-Golgi network. Proteins with the same target sequence are destined for delivery to the same location. The trans-Golgi network then buds off into vesicles. These vesicles then migrate to their target location. These locations include internal organelles, such as the lysosome, the digestive organelle of the cell. The vesicles can also be targeted to the cell membrane, where the targeted protein can be released from the cell for delivery elsewhere in the organism.
Well, that's great. That provides, oh yes, there we go. That provides, I think, really nice um, explanation of sort of how these organelles are involved in actually doing things, right? The rough ER sends materials to the Golgi. The Golgi activates those proteins, and then the Golgi releases these vesicles. Oh, let's stop there. The Golgi releases these vesicles. The process. Oh my gosh. Okay. Sorry, I'm going to back out of here and stop this madness. Again, the Golgi will go and package things up and then ship them to other parts of the cell. So now you have proteins delivered where it's needed to make the cell work. Okay, lysosomes. Lyse means to split or to break. So lysosomes are, you can think of them as like the recycling center of the cell. You can have old cell parts or organelles that are damaged. You can have proteins in the cell that may need reworking, and you can send them to the lysosomes. And inside the lysosomes are hydrolytic enzymes that will cut up and degrade those organelles. So you can actually recycle those parts. Okay. So again, lysosomes are recycling centers. Another cool thing that lysosomes can do, depending on the cell, they can take in or swallow up materials from outside the cell. And then those little vesicles that have, in this image, they show an engulfed particle here, they can fuse with the lysosome, and the lysosome can digest those particles. This is one way like sponges eat. Sponges will have little vacuoles of food particles suspended in water, and then lysosomes can go and digest that stuff. One instance of like when things don't work, there are a couple of enzymes, a couple of hydrolytic enzymes in lysosomes that digest lipids. And if those enzymes don't work, then what happens is an accumulation of those lipids in the central nervous system. And that's what leads to Tay Sachs disease. Kids with Tay Sachs, they're born healthy, but because their lysosomes lack the enzymes for breaking down lipids, the lipids will actually accumulate in their brain. And starting at age like 3 to 6 months, they start to have neural degeneration. And then, it's sad, but the kids all die by the time they're like 5 or 6. A person's lifespan will only be half a decade because of a couple of enzymes not working. So it's pretty profound, what lysosomes can do for us. Now, speaking about vacuoles in general, I like to think of vacuoles as being like little Ziploc bags for the cell. They're just little membranous sacs, and you can package up whatever you need in them, and you can ship that stuff all around the cell. Now, there are some really cool specialized vacuoles, like contractile vacuoles. Contractile vacuoles do exactly as the name suggests, they contract. Little protists like Euglena that live in fresh water, they've got a bit of an issue because water tends to flow into their cells, which will cause them to explode. So what they do is they have these contractile vacuoles that are sort of like a sump pump. Water flows in, the vacuole contracts, it squirts water out. They can actually help propel themselves along with these contractile vacuoles. Another vacuole that you're probably more familiar with is the central vacuole that's in plant cells. The central vacuole fills up with water. It can store all sorts of wastes and stuff like that in the plant cell. But what happens is when the water flows into that central vacuole, it pushes on the walls of the plant cell, and it helps the plant stand up against gravity. It's also what makes certain fruits and veggies really juicy. When a person bites into like an apple or a carrot, it's crisp because of the cellulose in the cell walls, and it's juicy because when you rip through those cell walls, you puncture the central vacuole, and the central vacuole releases all sorts of molecules. Okay. So again, central vacuoles store materials in plants, and that's what you're biting into, or is releasing juice when you bite into like a plum or a peach or whatever. All right, let's pause there, and then we'll get on to energy conversion.